Hey everybody, Mr. Bress here, and I just wanted to record a recap of today's lecture or class on Thursday, February 4th. Today we did at the beginning review the sort of monohybrid complete dominance crosses. And then at the end, I briefly touched on what the new content for today was, which I incorrectly called codominance. It's actually incomplete dominance. Codominance is what we'll learn about tomorrow, where both traits show up fully. In incomplete dominance, you get more of a blending. And I'd like to just quickly go over to notability and show an example of what I mean, and then go through the practice problems that you had today, just so you know that you're doing those correctly. So let's share my screen here and hop on over to notability. And we'll start with a pretty common, I'm just going to open up a new window here. Let's start with a pretty common example. So in the past, let's say there was a flower could be red or white. And up until this point, we might have seen that we might have seen something like red is dominant over white. So in this case, you would make, maybe let's make this a flower. So capital F would have been red, and lowercase would have been white. And in complete dominance, which is the original case we learned about, complete dominance, what we would see here is, if you are homozygous dominant, the flower would be red. If you are homozygous recessive, we talked about the flower would be white. And which was kind of new to us or interesting when we were learning about genetics in the first few days, is that this heterozygote, one of each, the heterozygous, mean one capital and one lowercase. Well, we found out that that would also have been red in the case of complete dominance, because there it only takes one of the dominant traits to give the organism that trait. Now that's great in complete dominance, and that's what we've been learning about. But there's another special case that comes up, not too many examples, especially not too many examples in introductory genetics, where you get incomplete dominance. And this is actually probably a little bit more like what you would expect prior to starting genetics. And in incomplete dominance, let's say once again, you have a red flower and a white flower. Okay. And let's say that capital R gives you red, and well, actually, that's not exactly. Let's back it up just a little bit. Let's say there are three possible genotypes: big R, little r, and big R, little r. Okay, and let's figure out what these equal. And so the top and the bottom, we might get, well, if it's two capital R's, and you might recognize that this would be red. It gives a red flower. And if there's two of the lowercase r's, you're like, okay, well, that would probably be the white flower. Now, the question is, what is the heterozygote in the case of incomplete dominance? So previously what we learned is that this would be red, right? And that is true in the previous case, but now in incomplete dominance, this is a little bit more like what you might have expected to begin with. This gives pink flowers, okay? And so you might imagine early geneticists who were experimenting on plants could have crossed a red flower with a white flower, and they would have done their Punnett square just like we had done in class, here and here, and they get their two red gametes and two white gametes. And then they would have looked and said, okay, I'm gonna get this allele here, get this one here, here, and here. And you might recognize that all of these here are 100% heterozygote. But then what they saw was, oh yeah, all of these flowers are pink. 100% are pink. So basically to them, they crossed here's my flower, a red flower crossed with a white flower, and they got all of the offspring gave them pink flowers. And they were like, okay, yeah, that makes sense in the world. Red and white, you mix it together, it's pink. They're like, the world makes sense. That's not that interesting. But then these early geneticists said, well, what if I take this pink flower and I cross it with another pink flower? What happens? Well, what they saw was they got 3 types of flowers back. They probably thought pink and pink can't get any more pink. It's just going to stay pink. But they got very distinct colors. They got red back. They got white back. And they got pink flowers back. Crossing 2 pink flowers, in this case, they got all 3 different kinds of colors flowers back. So how did that happen? And I'm just going to stick with black here. Well, the pink flowers, remember, are the heterozygote. Capital R, little r gave pink flowers. So these geneticists crossed 2 pink flowers together. So genetically, what does that look like? They went big R crossed, or sorry, big R, little r crossed with big R, little r. 2 heterozygotes crossed together. And when you do the Punnett square, you get big R, little r, and big R, little r. And then when you cross 2 heterozygotes, you get this very classic distribution. You get this 2 capitals, 2 lowercase, and 2 heterozygotes. And what we know up here was that big R is red, the heterozygotes are pink, and the little r were white. So this is exactly what they got here, is they got red flowers, they got white flowers, and they got half of the flowers were likely to be pink. So they got all 3 colors back from there. So this is the only difference in incomplete dominance versus complete dominance, what we saw before. So let's go, let's see if we can practice a couple of these problems without having too much going on here. So I'm going to go into Canvas, click on modules. I'm going to look at today, incomplete dominance, and I'm going to click on down here on these practice problems. And it comes up here. I'm actually just going to open up the PDF as a whole. I clicked up at the top there, and then I'm going to send this out to Notability. I'm going to create a new note, and we should have this in Notability. And let's go take a look. So back over to Notability, incomplete dominance. Okay, so here it says, I'll get my pen back at black. In Snapdragons, flower color is controlled by incomplete dominance. All right, the problem's telling us the 2 alleles are red and white. And the heterozygous genotype is pink. So what phenotype is the plant with the genotype RR? Well, this is red. What is the phenotype of the plant with genotype r? That is white. And the heterozygote is pink. Okay. A pink flower is crossed with a white flower. So I have a pink flower. Pink flower is crossed with a white flower. Okay. And so we know that this genotype will be, what is the probability of producing a pink flowered plant? Okay, oops, I think this parents, I was supposed to have 2 of those. Let's just cross that out. We don't need that. Okay, so a pink flower, so that is capital R, little r, is crossed with a white flower. And what is the probability of producing a pink plant? Well, let's see here. So these go here, big R, little r. These go here, little r, little r. And I see a couple pink plants at the bottom and a couple white plants. So I see 1, 2 pink plants and 2 white plants. So here, the probability of a pink plant is 2 out of 4, or 50%. So the probability of producing a pink plant is 50%. So now we're asking what cross will produce the most pink flowered plants? Let's cross this out. I think I had meant to do something else with that before. Well, I want to get, if I know I want pink plants, I want big R, little r. And I'll let you think about this so you can pause the video. And then now, when we come back, let's figure out how to get the most of these. Well, I know the way to get the most heterozygotes is to 2 capitals crossed with 2 lowercase. So this is red crossed with white, and we can prove it with a Punnett square here. These go here, big R, big R. These come in here, little r, little r. And you can see when you make the cross that 100%, or 4 out of 4 100% are pink. So that is the way, these are the offspring. These are the parents, a red plant and a white plant, that give the most pink plants. Okay, so what else do we have here? One more. In this beginning genetics, you really see snapdragons and these birds, these Andalusian fowls, that display this codominance. These are the ones that teachers always go back to. So in these birds, the individuals that are black are BB, and white are little b. And it's saying, okay, so let's just take a look at this. A homozygous black bird is crossed with a homozygous white bird. So let's just, I'll use the colors to begin with. So big B is crossed with little b. And all the offspring are bluish gray. Show the cross as well as the genotypes and phenotypes of the parents and offspring. So here, when we cross this, just like we did before, this is big B crossed with little b. Every single one of these is going to be heterozygote. And it's telling us that every single one of these is bluish gray. So here, big B, the heterozygote must be bluish gray. All right. What results if a black individual is crossed with a bluish gray individual? So here we say the black individual is big B. So big B is crossed with a bluish gray individual, which we found out from the previous problem is a heterozygote. So what does this look like? Well, once again, let's use our Punnett square, add in our gametes here, big B and little b. Sorry, I'm just keeping it in black this time. And what we see is two out of four, or 50%, will be black. And we see these two heterozygotes here. So 2 out of 4, 50% will be bluish gray. Okay? If two bluish gray individuals were crossed, what would be the ratios of the phenotype and genotype? So phenotype is just the color, and the genotype is the two letters. So let's take a look. Well, we're crossing two bluish gray individuals. Those are both heterozygotes. And you're probably getting pretty good now at maybe starting to guess or remember what that distribution is when you see two heterozygotes crossed. And it's always the same, and it always has those two capital B's at the top, or two capital letters at the top left, two lowercase letters at the bottom right, and then what I call these kind of in-betweens here are heterozygotes. So this individual would be black. This individual is white. And again, that's based on what we learned up here. And these two here, these two heterozygotes are going to be bluish gray. So what we have is we have BB equals 25%, because that's 1 out of 4. So 25% are black. 50%, because that is 2 out of 4. 2 out of 4 are this bluish gray. And finally, little b, 25%. Hey, how's it going? 1 out of 4 are white. Okay, so that is simple. I think if you understand the basics of the monohybrid cross, that these incomplete monohybrid cross with complete dominance, that's what we originally started with, that this here, this incomplete dominance, I think is going to be pretty easy for you to see, and it makes the problems a little bit more fun. So I'm going to add this video to today's Canvas page so you can use it to refer to, and I will see you all in class tomorrow. All right, have a good night.